Welcome to another episode of the Bleeding Edge of Digital Health. Really excited about today's guest, Mr. Peter Vranes, co-founder and CEO of a terribly exciting technology and company, Nutromics. Peter, how are you doing today?
Great, Mike, and really happy to be here, having a chat with you and
Yeah.
the viewers too.
Yeah. No, for the audience out there, got Peter after a 15-hour flight from Australia over here to the stateside. So I appreciate you being up bright and early to film this. And we've had a number of conversations. Met back in March at LSI, and obviously had a long chat there, and then we've had a couple conversations since, and couldn't have been more excited when you agreed to come on the show. This is a category of its own, a one of one at this point, and I think the audience is going to be really excited about the technology, as excited as I am. So we're excited to jump in here. You guys kind of term the technology a lab on a patch, and rather than me fuddle through trying to describe the technology or the category, I'll let you tell the audience how do you guys categorize this?
We're a diagnostic, Mike. We're a diagnostic company, and we've developing a new methodology to continuously monitor molecules in the body, basically. And this gives you, it's a patch, which you can see here. And the patch is made up of microneedles, and they're a bit hard to see, but you can see 4 little needles there. And on the end of those needles, we put a DNA-based sensor. This is the engine of our technology. And this is a platform technology that can monitor any diagnostic target continuously and in real time. And the way it works, we're starting off in the ICU, in the hospital. You pop it on, and there's a little mechanism. There's a integrated applicator, and you just pop that down. There's a little click, and those needles penetrate the skin. And those needles are only about 2 to 3 millimeters long, so you basically can't feel those. But what it does, it gives us access to interstitial fluids. That's the fluid just under the skin. And it's the same fluid that continuous glucose monitors use. And so that's what allows us to get access to the molecules that we're interested in measuring. And so where we're starting with this is measuring a drug called vancomycin. And it's the number 1 dosed antibiotic, very hard to dose today. There's a narrow therapeutic window. It's used for conditions such as sepsis, MRSA, so very fast-moving bacterial infections.
Sure.
And clinicians get very little data to dose the drug. And so by continuously monitoring that drug, vancomycin, we can help doctors to get patients in that therapeutic zone where they're killing the bacteria and keep them out of the toxic zone. So that's where we're going to start, but we're going to expand it because we can do multiplexing. We can do multiple sensors on the one patch. So we'll start there, but we'll expand it out to other indications after that.
That's incredible. Yeah, the vancomycin, we talked about that at LSI, and I did a little bit of research when I got back, and certainly want to get into that a little bit later on in the show here, because that's a challenge that the solution is almost as much of a problem as the original problem at times, and I think it really speaks to the value proposition of what you guys are creating. What was your background? You've got an illustrious background. You're a serial entrepreneur, multiple exits. What was the origin story for the technology? And I think one thing it's worth noting to the audience is I love it when people don't have a huge healthcare career and then they come into healthcare. It brings fresh perspective. You don't know what you just don't know, which it can oftentimes be extremely beneficial. But what was the origin of the story? What made you decide to get into healthcare? And obviously, you've got a co-founder. Would love to hear a little bit more about that.
Yeah, you're spot on, Mike. We both Hitesh, my co-founder, and I, neither of us came from healthcare. And it's interesting when you look at some of the biggest innovations in the world, they often come from people that are outside the industry. And I think there's a number of reasons for that, but one of them is that when you come from outside of an industry, you're not that you don't have a paradigm that you have when you're in an industry for 20 or 30 years. I think there's a certain paradigm that you have in that sort of situation that you don't have when you come to an industry fresh. There's pros and cons with that, obviously, because you also don't have certain experience as well. But yeah, so the origin story was I've been in skincare for many years. So I've gone full circle from new products to exits, and was starting a new skincare brand. I exited one company back in 2013, sold that, and started a new skincare brand. And part of that brand was working with a company in Germany on a technology that could monitor carotenoids in the skin. And this was a skin health brand. So carotenoids are an antioxidant, and antioxidants are an indicator of skin health. So we wanted to do a clinical trial looking at that. And at the time, I was reading a book by Tim Ferriss, and a lot of people would know him from the 4-Hour Workweek. But he's written a few books, and one that I read at the time was The 4-Hour Body. And I was getting into biohacking and looking at micronutrients, and asked the guys that I was dealing with in Germany, what else could this technology that they had measure? Because carotenoids is one thing, but could it measure other micronutrients? That was the start of Nutromics, because I thought, gee, wouldn't it be wonderful if we could have a wearable that could monitor our micronutrients continuously, and then we could, and we're all deficient in micronutrients, just about every single one of us, but we don't know it because we're not continuously monitoring. And in this book, Tim Ferriss did a blood test, found he was deficient in selenium, increased his consumption of Brazil nuts, which is high in selenium, and then his testosterone levels went 3x just through this one little shift. So that was kind of the motivation. I thought, wow, imagine if you could do that continuously. Long story short, soon realized that people don't really care that much about micronutrients. They just didn't seem, outside of a small biohacker community, didn't seem to be much demand for that, right? So this story is a story of pivots in the early days. And this is back in 2017. Met Hitesh. I'd been a single founder my whole career for 20-something years. Didn't want to start another business as a single founder. Wanted to explore the whole co-founder thing. Didn't have someone that immediately I thought, that's the person I want to bring on. Went to this bit of serendipity, went to a conference, lean startup conference, sat next to a guy. We got to talking about healthcare, and that guy was Hitesh Mehta. We had a couple of beers after the conference, met up once, and I said, hey, how would you like to join me on as a co-founder of this new company? Wow. And he said, okay. we don't really know what it is, but let's give it a crack, right? It sounds interesting. And so we went from this optical technology that maybe could measure micronutrients that no one actually ever really wanted, to then looking at glucose, because we knew that glucose was really important, and we know about continuous glucose monitors, but there's it's a 1 centimeter cannula that goes in, and it's expensive, and there's all these user experience issues. So we looked at sweat, because you can look at glucose in sweat. Right. Realized that was going to be really hard. Then we looked at glucose on a microneedle. So these were all the pivots we're making.
Sure. And
then landed, there's certain a lot of companies doing glucose. There's Dexcom, Abbott, Medtronic. Do we really want to go up against these multi-billion-dollar companies? It's a big red ocean.
Right.
And then we heard about this technology out of UCSB, University of California, Santa Barbara, and it just ticked all the boxes. A guy called Professor Kevin Plaxco is a leader in the biosensing field. Some people might know him. And he developed this technology that's a platform that can measure any diagnostic target continuously in real time. And we thought, wow, that sounds amazing.
Right, yeah, how'd you find that?
there's a researcher, a professor in Deakin in Victoria, where we're from. We're from Australia.
Yeah.
And she's the president of the International Aptema Association. Now, we didn't know what an aptema was. Never heard of it. Right? Remember, we're outside the industry here.
Yeah.
And so she said. So we met her, and she said, "You got to meet this guy. It's Kevin Plaxco. It seems like what you're looking for and what he's got is perfect." So we did a bit of research on Kevin, and he published a few papers, like about 10 papers on this tech, and showed it working, and it seemed too good to be true. We thought,
sure,
why hasn't anyone picked this up yet? Like, it's the holy grail of biosensing, is
right.
a platform technology that can measure anything. I mean, CGMs are a $10 billion per year revenue industry. Three companies have a enterprise value combined of $160 billion based on being able to measure one thing.
Yep.
So we literally jumped on a plane, and we landed in I think it was LA. I can't remember, LA or San Francisco. Jumped in an Uber because we didn't even know there was there was an airport. There's a tiny airport in Santa Barbara. We didn't know that.
Yeah, I went to school by there. Yeah.
No, did you? Yeah, 3-hour Uber ride, which had a shower in some gym. 3-hour Uber ride. We're jet lagged, and we're here to meet Kevin Plaxco. And we're sitting in the office, and all the walls moving because we're so
tired,
exhausted. We're trying to make a good impression,
Sure.
that we know what we're talking about. We didn't know anything. We're not from the industry,
right?
And we're trying to secure this technology that seems amazing, right? And we did that twice. We did it twice. So the first time we spoke to their tech transfer, and I think they said, "Yeah, come back maybe another time," and just like it's down the road, you just come back and have a chat to us another time. No worries. So the second time we got there, they said, "Look, we really love the grit that you guys are showing here. Like, you're sort of a bit of a dog with a bone here. You keep turning up, annoying us a little bit, trying to get this tech, but it's with another company, but what's going to happen is that they're using it for a slither of the field. So there's all these fields you could use it for, and they're just using it for brain implants. So the company in Baltimore, and they're going to send most of those fields back because they don't want to keep paying all the patent costs. But then we've promised it to another company when it comes back. So sorry guys, there's nothing we can do here. So we thought, boy, they could have told us that earlier, right? Like, there's two trips, a lot of effort. So we had an option. We said, well, we could either just go back, tail between the legs, and we tried and it didn't work, or it's a bit of a ballsy move here, but maybe we fly to Baltimore, have a chat with the guys who are going to send it back, and see if we can do a deal before it comes back. Because we know if it came back, there was no chance we're going to get this tech, right? So we thought, well, what have we got to lose, right? So we literally jumped on a plane, went to Baltimore, had a chat with these guys and said, hey, we're really keen, right? We're really, really keen, and we want to work with you. You guys know this technology really well. We want to learn it, and we'll do some consulting with you, and we'll do some, but we want to sub-license that tech.
Yeah.
And they said, look, give us 5 minutes, and they stepped out of the room, came back, and said, all right, you got it.
No
and it was
way.
That moment literally was the making of Nutromics. That was
5 minutes, and they said we're in, huh?
You're in, and so, and it was one of those moments where you can't go high-fiving in front of them, right?
Right.
It probably wouldn't look good,
right?
But we got around the corner.
You knew you had what you needed,
and this was like, holy shit, we got it! Like
that
it was literally
awesome.
It was because we knew what it meant, right? We just knew how important it was going to be to us.
Yeah.
And then we hoped the technology would be everything that we expected it to be, right? And that was February 2020, just before the pandemic.
Right before the pandemic, a month.
Literally right before. And so we came back to Melbourne, and I think there was maybe 4 of us at the time, 3 or 4, or something like that. Today, there's 55 where we've got a decent sized R&D facility in Melbourne, Australia. I'm calling from San Diego. We have a small R&D presence here. We're about 6 employees now in the US. And we Dexcom's one of our major investors, and it just changed our world. Because of what we could do with that tech. We've gone and built the technology out. We've got a fully integrated working prototype that's now in clinical trial. We've got this device that we're going to be taking to the market. Our world changed at that moment, and that's all happened in 3 and a bit years. So it's gone. That was like the once we got that, the pedal went to the metal, and our world changed. And I'm just, it's one of those moments where, boy, it could have gone. There are just decisions that you make that when you look back, it could have gone. We could have easily just said, "Oh, well,
you could have got back on a plane and gone home."
Yeah, we tried and it didn't work, and let's do something. Do something else. But it's those moments that are pivotal that can change your world, right? So that's the origin story.
Yeah, I love it, man. I love it. I mean, there's so much. You could autopsy that process, and so many, inflection points, and like we've talked about in the past, having that grit of like, no doesn't mean no. No just means not right now, and I'm gonna figure out a way to make this work, right? I love that. I
would say, like, we always like talking about principles, because we run the company based on certain principles, and grit's one of them for sure. Before we were talking about the book Grit, that's a great book. I've given it to my son actually, and he's actually reading it. Sometimes I'm not sure if I buy him a book if it gets read, but he's reading that one, which is great. But that was certainly, grit was part of it. The other one was, and this is in our DNA as a company, we've always run by this principle, is collaboration. So I talked about the pivots in the early days.
Sure.
The reasons we made quick pivots was because we just said, who is the best at X? So if you're coming into an industry fresh and new, you got to have a steep learning curve. That's a must.
Right.
So because yes, you don't have the paradigms, and that's that you have a freedom to say let's not go by. Let's not limit ourselves by certain paradigms. You have that, and that's great. On the flip side, you've got to learn really fast.
Yep.
And the best way that we've done that is that we've just said who's the best, and then we talk to them, and that's how we've learnt really fast. And that's why we pivoted, because we've spoken to the people that know what they're talking about, and they've given us really good advice, and we've spoken to multiple people.
Sure.
So we try and get a bit of a consensus, and that's that story that I just told you was foundational on those two things. Just never giving up, always trying to look for a way, but also just speaking to the world leaders in certain areas and learning from them, because you learn fast and you learn what's not going to work and what hopefully will. So they're the kind of two principles at play there.
I think I wholeheartedly agree. Is Dr. Plexco still involved in the company?
He is. Dr. Plexco now spends probably 3 or 4 months of the year with us in Melbourne. So,
oh, wow.
I just did a 7-day hike in Tassie. We tented together, so we know each other really well.
Fantastic. That's great.
He's a friend. He's a great friend, and it's we're just very like-minded, and I love spending time. He's a great guy,
Yeah, that's the best part of a startup, is when it becomes a family, right? And there's not only professional respect, but personal respect. Makes it all worthwhile for sure. What in terms of use cases? I mean, I know some of this stuff is confidential at this point, but maybe talk just real quickly about the critical aspects of vancomycin, because as you and I had talked about previously, the mismanagement of the application of vancomycin can kill a significant number of people. The only reason that they continue to do it or does kill a significant number of people, the only reason they continue to do it is because if they didn't, more people would die of
That's
MRSA and the other bacteria. And then if there's any indications that you can give us that you guys are pursuing, or any other information about that, because the way I look at this thing is you guys are you're really only constrained by the size of how many needles you can put on the patch, right?
That's right.
Ultimately,
it's the
build a
only
bigger patch if
technology-
you need to.
But you're spot on. Yeah, we can multiplex, right? That's a big deal. CGMs, they can't multiplex. They can put one needle on a patch, and it's because if you put another needle, you're measuring a reaction, and they confound each other. And that's why CGMs have never gone beyond glucose. It's pretty much that's all you have, right? But having said that, it's $10 billion per year revenue market. So it's a blockbuster market.
Right. Big enough to stand up on its own.
Yeah, but when you project out into the future, there are other really valuable targets that you can measure. And the reality is that patients aren't going to wear 20 patches, right? It just becomes, or 5 patches, or it just becomes impractical.
Right. It's like wearing 2 different watches with, you're just not going to do it.
Yeah, exactly. So the future belongs to platforms for what we're talking about here. So being the only technology in the space that can multiplex is a big, huge advantage. And it begs the question then, what are you gonna measure? So we've done a lot of work looking at that, because all roads lead through the first product. If we don't get that right, there is no second product. You don't get many shots on goal here. You've got to get it right. So we've done a lot of work on making sure our first market's right. So therapeutic drug monitoring of vancomycin, there is a pull from doctors to solve this problem. It's a big problem, and there's a huge pull. And I'll give you some stats. So this is a drug that is renally cleared, so cleared through the kidneys. Mm-hmm. Half of, and as I mentioned before, it's used for life-threatening bacterial infections like sepsis that moves really quickly. Half of all doses don't hit this therapeutic range. So to kill the bacteria, the vancomycin needs to be at a concentration in this range. If it's outside the concentration, it's not therapeutic.
It's not valid.
Nothing. Yeah. So you've got sepsis being untreated, and that'll kill you. Half of all doses are not in the therapeutic range. So that's problem number one.
So that's just wasted money.
Well, highly inefficient cost-wise, but
right.
From a patient perspective,
right.
You get there are 12 million people that die every year from sepsis. So when you're in hospital, you're not treating the bacterial infection, people die. Yeah. Now they die from underdosing of vancomycin. No one writes we didn't dose vancomycin correctly.
Right.
They died from sepsis. That's how it gets coded. But we know when half of the doses don't hit the therapeutic range, there's a lot of people dying because they're not getting dosed with the drug properly. So that's problem number one. Problem number two is that 43% of adults in the acute setting get a vancomycin-induced acute kidney injury. What that means is that they got now too much of the drug.
Right.
So they get an acute kidney injury. And acute kidney injury is one of the top 10 killers in hospital. It's highly toxic drug. 100,000 people in the US go into hospital with a bacterial infection and die from vancomycin-induced acute kidney injuries.
Wow.
So 100,000 people every year die not because of the bacterial infection, they die because of the treatment. It's an amazing statistic.
Wow.
When you really think about that.
Yeah, you marriage that up to the number of people that die died from COVID, I mean, obviously we're talking, but I mean, it's relevant. Yeah. It's not like we're talking, 100, 200. That's a significant population. It's a huge number.
Then there are a lot of people that don't die. They get an acute kidney injury, they survive, but it's one of the major predictors of chronic kidney disease.
Yep.
So it puts them on a path of chronic kidney disease. There's about 800 million people in the world with chronic kidney disease, double the number that have diabetes. It's a huge problem. It doesn't get spoken about enough.
Yep. My dad's got it.
Yeah. And that leads to dialysis and all the problems associated with that. So it's a huge problem, and doctors are dosing this drug every day. But if they don't use the drug, a whole lot more people are going to die because of the bacterial infection. They have to treat the bacterial infection. It's life-threatening. So it's the lesser of two evils. And the biggest problem is that if you go in a hospital and you need to get vancomycin, they'll basically have an estimate of your weight, and they'll give you an initial dose based on that, and then they'll take a blood draw, and they'll try and have a look at what concentration of vancomycin is in the blood, and then that'll be hours later, because they'll take the blood draw, go to the lab, come back hours later, and so they're always looking in the rearview mirror with very little data. So this is actually the heart. This is
the crux of the issue.
This is the crux of the issue. And so they say, okay, up it a little bit more, put it down a little bit more. It's a lot of trial and
guessing
error
game
to try and get. It's like trying to throw a dart blindfolded at a dartboard, right? It's hit a bit up a bit left, a bit more right. It's hit and miss, and it's what it is. And the guidelines call for a peak and a trough, 2 blood draws. 80% of hospitals don't follow the guidelines because it's too impractical. They haven't got the bandwidth to be able to do all the testing that they need to do. There's a whole lot of problems. So you can start to see why this is a low-hanging fruit for us.
Sure.
So putting a vancomycin sensor on this patch, this is the alternative, right? So a patient gets prescribed vancomycin. Doctor puts a patch on, or the nurse.
They see it in real time.
They see exactly where that patient is in real time. So their ability to get that patient quickly into that therapeutic zone where it's killing the bacterial infection and keep them there is greatly enhanced. Obviously, it's day and night.
Yeah.
So that's where we're going to start, right? And we know it's the number one dosed antibiotic in the world. It's a huge market for it. And there's a huge impact we can have on patient outcomes. That's the big driver for us, patient outcomes. But like we talked about, we can multiplex. So how are we going to roll this out? And so our vision for this is a patch for every patient. So that means every patient that goes in the hospital, they get a patch, right?
Yep.
That's a big vision. And the reason we think that vision it can become a reality is that we can put many very, very high value targets onto this patch. We can put 20 to 30. There's no limits. It's limited by the geography and the size of the patch. And when you can do that, then regardless of the path that the patient goes through the hospital, from triaging to drugs to early diagnosis of disease states to remote patient monitoring, we have clinical utility. So because of the targets that we're monitoring continuously, and so we think that that the clinical utility combined with the cost that we can pull out, because taking blood draws and sending them to labs is highly inefficient and highly costly. We can pull out billions of dollars. We can get better patient outcomes. For hospitals that have patients are in hospital less, in the ICU less, which is where it's extremely expensive, less readmissions, and so we can pull out billions of dollars out of the hospital system with that technology. So that's where we're heading, and so we're starting with therapeutic drug monitoring. We talked about vancomycin, but there's about 40 drugs indicated for therapeutic drug monitoring. Then we add on targets for renal targets, because doctors are always interested in the kidneys when you're dosing those drugs, because most of them are renally cleared. Then we put in targets for sepsis, because the number one indication for most of those drugs is sepsis. And then we put in targets, some other real high value targets, for example, troponin for heart attack diagnosis, so cardiovascular markers.
That's a huge one,
And that's a huge one in and of itself. But that's how we're going to layer this on. And we've got a pipeline starting with vancomycin, where we're strategically developing these so that as we launch vancomycin, soon thereafter we've got product number 2, product number 3. And by the way, all of those products are on the same device. So the investment in the first one, all we're doing is putting another needle, another microneedle and sensor, and that's another indication on the same platform.
It's unbelievable. So really, a lot of the product development costs are baked. They're, you're just at that point you're just going after additional indications, but for the most part you've got fixed costs as it relates to new technology or the actual form factor development.
From that perspective, yeah, from the device. So there's incremental costs from a regulatory perspective.
Sure.
So there are some incremental costs, but compared to what normally medtech companies would have to incur for new products, 80% of it is we're leveraging 80% of it over and over again because we're not creating that again. That's created once, and we're just adding, as I mentioned, another needle, another sensor.
Yeah. No, that's incredible. What the troponin piece is really interesting. But that's the first thing they do when somebody comes in with chest pain, right? They
correct. Yeah.
Hook them up to an EKG and check their troponin levels.
Yeah. So there's 10 million people in the US go through what's called the chest pain protocol. So someone's at home, they're at work, and they've got chest pain. They call an ambulance. Ambulance turns up. And they, like exactly what you said, they put the leads on, they send them to hospital. And the guidelines for MI for a heart attack is a rise or fall pattern of this protein called troponin. And so when you have a heart attack, you get there's a release of this troponin, a real stark increase or decrease. Depends. The reason I say decrease is because it depends when you get them.
Right. If they've had it for a while.
Yeah, so if straight away there's a big increase, but then there's a decrease, and so the guidelines call for a rise or fall pattern in this troponin, but pattern is the key word here. So that means you need 2 data points,
right?
And so what they do, you go in the hospital, they take a blood draw, they send it to the lab, they get their 1 data point. Then they wait an hour, then they take another blood draw, send it to the lab. Now the lab doesn't turn these around in 5 minutes,
right?
So there's a time period because there's queued up with all the other things they've got to do. Average time for heart attack diagnosis in the US is 5 to 6 hours. Now, in that time, the heart is occluded if it is a heart attack, and heart cells are dying.
Yep.
And that's
the heart muscle is dying. Sure. Yeah.
The heart muscle is dying and doesn't get regenerated. So doctors would call that time is muscle. You may have heard the term.
Yep.
And so that's a big problem. Every minute is precious when someone's having a heart attack. Now, 80% of people that go through the chest pain protocol are not having a heart attack. They've got some other issue going on.
Yep.
There's
angina or whatever.
Could be lots of different things. But they're going through all this process because that's the process. So what we're working on is a troponin sensor. So on this patch. So let's have a look at another alternative here. Ambulance turns up, someone's got chest pain. First thing they do, they pop on a patch. And we're streaming troponin immediately, continuously and in real time. Now we can see that rise or fall pattern in 5 to 10 minutes because we're taking, we could take a reading a second, a reading a minute, a reading every 5 minutes, depends on the application. We could do sub-second concentrations if the application called for it. So that means that within 5 to 10 minutes, they will see there's a rise or a fall pattern straight away. And that's critical because then they say, hey, you're in that 20% that you're actually having a heart attack.
Yeah.
We're going to send you to the nearest cath lab. Prep the cath lab straight away.
I was going to say the cath lab's already ready. They've got the case set up. The room is ready. Wheel the patient in. Away they go.
Yep. So and that time is muscle. That's critical. Or you're not having a heart attack. You can relax. We're going to work you up for something else. There's some other issue, but it's not a heart attack.
Yeah.
So that's a game changer for one of the greatest burdens in medical burdens we have in the US and around the world, cardiovascular disease. That'll be a huge shift. And even just, I know personally, my sister had a heart attack about a year ago, and she flatlined in the hospital, and they brought her back, and then they send you home, and every little tweak, little, you're thinking, oh my God, is this happening again? So even from the patient perspective of just, she said, gee, if I had that, if I was wearing a device that I knew I'm not having that little tweak is so fine. It's not a heart attack. That would give me enormous comfort. My anxiety level comes straight down. So there's multiple levels of benefit. Rather than, it's not just the hardcore diagnostic benefit, which is important, but there's certainly a patient
peace of mind.
Benefit as well that comes with that.
It's incredible. Yeah, I mean, obviously the vancomycin, as you said, is the obvious indication, but man, having sold coronary products, coronary stents early on in my career, that would be an absolute game changer. And as I kind of look downrange with that, I don't know if there's any tests that they do for ischemic stroke, but if there's any biometric indicators for ischemic stroke, but that's another one where time is brain. They have to get the clot out, so that there may be something there as well. I want to be sensitive to time here, but I know the audience is going to be interested in two things. One, is there anything else in development out there like this? And then two, what's the projected, do you guys have a target date? Obviously that's always fluid, but do you have a target date for when it would be released for vancomycin?
Yeah, I'll answer the second question first. Late 2027, early 2028 is our launch date.
Yep.
There's we've got to go through the FDA process. We're in clinical trials at the moment. Got our pivotal trial during that, leading up to that. So, but that's about the time frame we'll have this in market. In terms of competition, the major competition really is CGM. It's based on an enzymatic sensing technology, and the innovation we're seeing in that space is around using microneedles instead of so it's typical CGMs have you have a big applicator, you have a cannula.
It's a cannula,
and there's a film. Yeah, and it's up to 1 centimeter. Some of them are smaller, and so because it's longer, there's certain places in the body you have to put it because you can't just put it anywhere because you need fatty tissue to be able to allow the cannula to go in, and so we're seeing starting to see some innovation in just smaller needles in that space. But in terms of a platform technology that can monitor any diagnostic target, we are unique in that respect. And the interesting thing was that I didn't mention it before, but I find it fascinating that biomimetics. So biomimetics is learning from nature. How did nature solve a problem and then learning from that? And so Kevin Plaxco, the inventor that I mentioned earlier, he's a biological physicist. And in your body and in my body and all of our bodies, we have proteins that change shape in the presence of their target. That was his light bulb moment. And so he thought, well, instead of using proteins that are very difficult to manufacture, I can use these things called aptamers, DNA sensors basically.
Sure.
But it does exactly the same thing. It changes shape in the presence of its target. And that was the learning from nature light bulb moment that actually is what started him off on this direction, 10 years ago or so. So, and that's unique. That conformational change for sensing is very, very unique. And there's a paradigm shift in the sensing world where it's before enzymatic sensing is measuring a reaction. This is measuring directly the target through conformational change, and there's a signal change that we can measure. So to answer your question, there isn't a technology that is similar to that because it is so unique. But and we are really pioneering that. We're the world leader in continuous molecular monitoring. As a company, and so we're really pushing that ourselves.
It's quite a tremendous opportunity, not just in vancomycin, but all the other exciting things going on out there. And it's obviously there's a financial consideration there, but man, the impact on patients that this is going to have is it's almost immeasurable, really. It's pretty unique. So Peter, one of the things that I always get questions on is, how can people get in touch? Because people are going to see this episode and they're going to say, I want to work for that company. I want to work for that guy. And for those that are out there that are listening, I've had multiple conversations with Peter at length, and I can tell you he's every bit as good of an individual as he appears on this episode, and you're going to get a lot of people that want to work for the company. What if they're interested in in a career at Nutromics? What's the what would be the appropriate way to reach out? Just go to the website, or?
Yeah, we have. We actually have a new updated website where we're putting all the job roles that are open at the moment. So that'll be, I think it's up now or just about to get up. So that's a good place to go. Look out on LinkedIn for jobs that we post. Connect with me on LinkedIn, because I'll always amplify the job post myself personally, and so typically you're always going to find out. But just on that point you brought up, Mike, our belief at our culture is that to revolutionize an industry, and that's a big claim to make, because that's what we're going after. We're going to revolutionize diagnostics with this technology. You need two things. You need a great tech, and we talked a lot about the tech, but you just brought up a really, really important point. You need great people.
Yeah.
And we and that's easy to say, but the question is, well, what do you do to demonstrate that? And so we haven't got time to go through all of it. Maybe it's another segment. But
yeah, I'd love to have you back.
But it's a huge part of what we do is looking after our people. We say of all the stakeholders we have, the number one stakeholder, and this is a deliberate approach, is our team.
Yep.
Is our people. They're our number one stakeholder, and that's how we serve the patient best, through an engaged, passionate workforce that love what they do. They love the vision. We look after each other. So yeah, so we're very, if there's people out there that just this strikes a chord, I think about 15% of our workforce now is from the US. We have a facility here in San Diego, but a lot do come to Australia because they love the lifestyle there, and why not? If you don't mind the scary spiders and snakes and stuff like that, which isn't where we are, by the way. It's more up north, but we are very, very keen to always talk to people who are really passionate about our vision of a world of zero preventable deaths due to a lack of continuous molecular diagnostics. So yeah, thank you for bringing that up, and thanks for having me on the show, Mike. I really appreciate it.
No, it was my pleasure, and but you got to promise to come back as we get a little bit closer to FDA launch. I want to get you back on here and do an update, and really appreciate you taking the time today. That's it for this edition of The Bleeding Edge of Digital Health. Thanks for tuning in, and we'll catch you next time.
Thanks, Mike.